Today, our team, Siti Hanani, Syaida, and Alia Izzati, will present our BDA Lab 8 project, predicting iris flower species using machine learning. Our core objective is to perform data analytics and train a machine learning model to accurately classify iris flower species based on morphometric measurements of their sepals and petals. Why the iris dataset? Introduced by Ronald Fisher back in 1936, it remains a classic benchmark in pattern recognitions and multi-class classifications. Its low dimensionality, clean numeric features, and perfectly balanced classes make it an ideal baseline to evaluate classification performance. Looking at the dataset structure, we have 150 total records containing four continuous numeric features: sepal length, sepal width, petal length, and petal width, and one categorical target classes. In terms of data health, there are zero missing value across all columns, and the classes are perfectly balanced with exactly 50 instances per species. This ensures our models won't develop bias to any majority class. During exploratory data analysis, we inspected dataset summaries and generated a pair plot to visualize feature relationship across species. As shown in our execution output, the data types are clean float values with balanced 50 sample distributions for setosa, versicolor, and virginica. From our visual analytic, three key insights emerge. Setosa is linearly separable, forming a completely isolated cluster in petal plots. Petals are stronger predictor than sepals, displaying a strong positive correlation between length and width. Versicolor and virginica show slight class overlap, demonstrating why a machine learning classifiers such as K and N is needed to draw precise nonlinear decision boundaries.
First, the dataset was divided into two parts. The four measurements, which are sepal length, sepal width, petal length, and petal width, were used as the input features or X. The flower species was used as the target variable or Y. Next, the dataset was split into 80% training data and 20% testing data using the train test split function. We chose a 80-20 split because it gives the model enough data to learn while keeping some unseen data to test its performance fairly. Since the dataset has 150 samples, the calculation is 150 times 80% equal to 120 training samples and 150 times 20% equal to 30 testing samples. We also use stratify equal to y to make sure all three flower species have the same proportion in both the training and testing sets. So the training set has 40 samples from each species, and the testing set has 10 samples from each species. Finally, we selected the k nearest neighbors or KNN algorithm with k equal to 3. KNN predicts the flower species by finding the three nearest flowers in the training data and choosing the species that appears the most. We chose KNN because it is simple, works well with numerical data, and is suitable for the iris dataset, where the flower species are clearly grouped.
Our KNN model achieved 100% accuracy on the 30 sample test set. The confusion matrix shows a perfect diagonal, zero misclassifications across all three species, including versicolor and virginica, which overlap on sepal measurements in our earlier EDA. Accuracy is the percentage of total correct predictions. Precision tells us of everything the model predicted as a species, how many actually were that species, meaning no false positives. Recall tells us of everything that is that species, how many did we catch, meaning no false negatives. The model performs this well because petal length and width create tightly separated clusters per species, so the three nearest neighbors almost always belong to the correct class. In conclusion, here are our key takeaways for this project. First, perfect test accuracy. We built a k nearest neighbors classifier with k equals 3, and it predicted iris species from sepal and petal measurements with 100% accuracy on unseen test data. Second, petals drive the prediction. Petal length and width turn out to be the strongest predictors, showing the clearer separation between species and cleanly isolating setosa in particular. Third, sepals are less reliable. Sepal measurements alone overlap significantly between versicolor and virginica, so the model needed the petal features to properly resolve those two species. And finally, this shows that simple models can produce strong results. KNN suited this task well because the dataset is small, balanced, and low dimensional, a good reminder that model complexity isn't always necessary for excellent performance. That wraps up our project, a machine learning model that reliably predicts iris species using morphometric measurements, with petal dimensions doing most of the heavy lifting. Thank you.